This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1263, recorded on October 15th, 2025. I guess it's the Ides today, the Ides of October. Is there such a thing? This is the Ides of March. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. See, everyone is really good. They're not answering me because they know they have to be introduced first. What did I say? I've forgotten what I said because I'm just talking. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 68 degrees Fahrenheit and a nice sunny day here in Madison on October 15th, which I have never before heard called the Ides of October, but you know what? I'll go with it.
I just know the Ides of March are the 15th, right?
Yeah.
But maybe it's not. It's 20 C here, partly cloudy, although it looks like the sun's going to come out at 3 p.m. here on the thing. Did you get all that rain over the weekend?
So much rain.
Yeah, it was
yes,
miserable.
so much.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is palindromic today here in Western Mass, 61 Fahrenheit, 16 C.
Wow.
And it's cloudy. It's been overcast ever since we got the deluge this weekend.
You got it too up there?
Oh, we got poured on. Yeah.
And from Montreal, Canada, Angela Mingarelli.
Hello, everyone. So we did not get this deluge you speak of. It was fine. I guess it missed us, but it is 55 Fahrenheit here, so that's like 13 C. So it's the coldest, but expected.
But it's not bad. It's not bad still.
It's not bad. And you should be happy you didn't get that rain. It was a lot.
Yeah.
We have lots of snow in our future. So.
Yes.
It was also a lot of wind.
Mm-hmm.
Somewhere I saw a news clip. There was it was a parking lot or station with solar panels, and the wind blew one of them off, and it hit a pedestrian and killed him.
Ooh.
Whoa.
Doesn't that suck?
Yeah.
Like you're walking to work and you get killed. I
Wow.
mean, well, I know people die every second, but that seems like a
Freak
waste.
accidents can happen. Yeah.
Yeah, freak accidents.
If it's not a solar panel, it's a light pole or all sorts of things blow loose.
Because our society is very complicated.
Yes.
Mm-hmm.
Of course, if you're living in a cave person society, right, hunter-gatherer, there are other things that can claim your life.
Infectious disease.
Infectious
disease, big one, but
Yeah.
also
But maybe-
predators,
Wild animals.
Mm-hmm.
wild animals, So I'd rather have the technology, I suppose. Then we could do podcasts. You couldn't do podcasts from a
You could not.
hunter-gatherer.
You could not.
No.
Unless you had people gather around, all 100 people in your little clan could
Which
gather
is what
around.
which is what one used to do. I mean, that was entertainment.
That's true. That's true.
I know, but it doesn't seem like it would be very comfortable. You'd probably be cold. It might rain on you.
You'd be used to it. You would have been doing it since you were little.
Well, also the beasts might disrupt it, right?
Yeah.
Because they know that you're gathering in large numbers and they could pick a few of you off. So, well, if you enjoy these programs, we would love your support to support them. We don't do ads, as you know. And so we depend on listener donations. And if you go to microbe.tv/contribute, we have a variety of ways for you to contribute, including we sometimes do live events. The last live event we did was in April 2023, TWiV 1000, when everyone came to New York City. And now we're going to have a live event here at the incubator in December. It's going to be Immune 100. It's 2 orders of magnitude less, but still celebratable.
It's exciting.
December 22nd.
Wait,
Right
two
here at
orders
the-
of magnitude less?
I think
10.
it's only one.
Well, if you go from 10, yeah, you're right. From 100 to 1,000 is
Yeah.
just 1. It sounded better at 2.
It sounded good, but I was like, wait a minute. How many
I was,
episodes have we done?
I was going at 10. That's why. We've only done 10 episodes of Immune. December 22nd, 6 p.m. Doors open at 5:30. We will have the Immune team entirely, right? Me and Brianne and Cindy and Steph. We will have 2 guests, local guests. And if you're local and you want to come, we'll put a link to the Eventbrite page, or you can go to the homepage of microbe.tv. There's a link right up there and buy tickets. You have to buy tickets, and there are different levels. It's part of our fall fundraising, our holiday fundraising. There are different levels of tickets. There's like basophil, and there's macrophage, and there's T cell and dendritic cell and B cells. B cell is the highest one. I didn't do it. My social media.
I was going to say, this is the first time hearing of these levels, and I wonder about, whether all of the members of the team would agree on the hierarchy.
Yeah,
I'd say T cell. T cell's number one, in my opinion.
Each level has a difference. We're going to give you stuff when you come. So, they all get admission, but you get stickers. Everybody's going to get refreshments. You're going to get stickers. You're going to get a postcard. You're going to get a mug. You're going to get a poster signed by the participants.
And depending on the level, you get different antigens given to you.
Yes. T-shirt. Anyway, go to the link if you're in New York City. We've sold 3 tickets already.
And if you're not in New York City, then make travel plans.
Yeah, if you're not here, you can still donate. There's a link on the page where you can support us offline. But I think it will be live streamed. The incubator is not very big.
Well, I mean, you could go there. You don't have to be in New York City to get there.
It's true. There are many forms of transportation.
Many forms of transportation.
You could, but remember, we only have 25 seats, so you got to buy a ticket.
Buy a ticket soon.
And because it's we don't want to rent the theater and get into all that stuff again. Because I vowed never to do it again.
Yeah, I was going to say, ask Vincent how much he enjoyed that.
Yeah.
It's just that things didn't work right, I mean, in the end, it was great, but there were some technical problems. And so I don't want to deal with that again. But doing it here, well, there'll be other problems associated with it.
Where are people going to sit there, Vincent? Is it going to be outside?
No, it's going to be the other part.
Okay.
We're going to take out the table, and there's room. So the 6 of us will sit in the front on chairs. We're not going to have a table in front of us. And then we can put 5 rows of 5 chairs easily and back and a few on the side. And then I'm going to have Chris Kandayan come and do the recording. And I believe it should be live streamable.
Cool.
So that people can watch. Anyway, that's cool. That's a lot of fun. A couple of other news items. I have an article here from Time Magazine. CDC not functional after Trump administration orders mass firings. As you know, many people were let go last Friday. They do it on Friday, right? So they escape the news cycle, I guess. And it turns out that many of them were a mistake, so they rehired them because, I mean, can you imagine not doing your homework and seeing exactly who you're firing? Oh, no, that would take too much work.
And they fired like 700 people in excess. It's not even like it was 2 or 3 people.
No.
It was hundreds of people that were misfired and then rehired, from what I understand, the next day.
It's just like the Doge stupidity where they would just algorithmically fire an entire list of people and then go back and say, whoa, we needed a few hundred of them. So
crazy.
I mean, that's just amateur, right?
It's amateur, and it's incredibly destructive because, a lot of the time when you fire people, they go look for other jobs.
Mm-hmm.
And
yes, exactly right.
Even if you get right back to them, maybe they continue looking for other jobs.
Yeah, I can't imagine that people are in these positions, even if they got hired back, are making long-term plans.
No.
So it turned out that they had gotten rid of Morbidity and Mortality Weekly Report, which when I heard that, I said, this is terrible, right? Nobody's going to get any information anymore. Now, they've since restored them. They also fired 70 Epidemic Intelligence Service officers. These are people who track down diseases. And I have a former student who is an EIS officer. I don't know. Did you know Julie Allen?
Oh, I met her.
Yeah. I don't know. So I don't know if they were rehired, or they should be rehired because you need them.
If anyone is not clear on what an EIS officer does, in Contagion, this would be the Kate Winslet character.
Yes. Yeah, these are the people who show up at restaurant A and figure out where the pathogen came from.
Yeah. Anyway, I think it's, it's part of the destruction of public health by this administration, but it also illustrates, as if you didn't know already, how incompetent they are. Just ridiculous. Who put in the measles case?
I
Alan?
did.
Go ahead.
So this is just an update on something we've talked about before. I'm linking to an NPR story. You can find others online about it. Measles cases are continuing to climb, and now we're up to how many states?
Too many?
Yeah, too many. It's numerous states, and yeah.
It's probably, and as Sof it says here, it's probably more cases than we're diagnosing, right?
Mm-hmm.
Many more. So why do you think this outbreak continues, Alan? Or any of you?
It's almost as if we don't have an effective vaccine.
Huh.
Or it's almost like we're not using it for some reason.
Oh, maybe that's it. Maybe that's it.
We're back to hanging around the fire without vaccines. The infectious disease is just taking over.
Yes.
Having vaccines, we're just not using them because we're too smart, I guess.
So there are little pockets
I think
of
it's the
people
opposite
who are,
of that.
Yeah, I'm joking. I'm joking.
there are pockets of people who are not vaccinating their kids, right? Because they're not getting encouragement from the U.S. government, the CDC, and so forth. And so more and more parents are saying, "Oh, I don't need to do this."
Well,
and because those pockets, are a bit isolated from others, they don't necessarily mix. The percent of people who are unvaccinated in some of those communities gets really high.
Yeah.
And I suspect some of this now with CDC not only getting gutted but also not making recommendations is not even parents who have deliberately chosen not to get a measles vaccine, but maybe ones who didn't realize that they needed to get their kid this because they weren't told, or because their insurance company isn't covering it, or whatever their issues that come up here.
Yeah. Now, normally, if in an outbreak like this, the CDC and NIH,
An EIS officer would show up.
Yeah, and they would push vaccination. They would set up vaccine kiosks there and push vaccination. But RFK Jr., the head of Health and Human Services, has never once said to get vaccinated against measles, nor has he pushed it. And so that's why this outbreak continues. You got a big one up in Canada too, Angela.
We do. Unfortunately, we do. Not in Quebec. I think it's mostly in Ontario, isolated to Ontario. But yeah.
I think also in, what's the province where Calgary is in?
Alberta.
Alberta, right.
Mm-hmm.
I think that. And mostly in people who don't vaccinate, like Mennonites and
exactly.
others.
Amish.
But still, it's totally preventable. There's no reason for this to happen. So these kids are at risk, of course, for severe disease, but also their immune memory is being wiped out.
Their immune memory is wiped out, and they have that risk of long-term SSPE later.
Yeah, that's right. Yes, and in 10 years, we'll see a cluster of SSPE arising as a consequence of this. So it's very sad. All right. We will do now some papers to make you happier. Because science goes on and science is fun. Science is what makes your life better and keeps you healthy.
We're going to talk about some of good parts of vaccines now.
Yes.
Oh, yes. They both have to do with
They're
vaccines.
both about vaccination. One about a vaccine we're trying to get, and one about one we already have.
Cell reports interplay of host and viral genetic variations in modulating antibody responses to genotype 3A hepatitis C virus, implications for vaccine design. Alan?
Yep. The author's first author is Zhiqing Wang, and senior author is Azim Ansari, not to be confused with Aziz Ansari, the comedian. This group is from University of Oxford, University of Birmingham, University of Nottingham, Nottingham University Hospitals Trust, Liverpool School of Tropical Medicine, and Oxford University NHS Trusts. So it's an all-England team.
So we last talked about hepatitis C. Actually, in Montreal at ASV, we had Charlie Rice as our guest, who has done important work on hep C, and he talked about it. He talked about research leading to the ability to make antiviral drugs, which can successfully eliminate infection. There's no latent reservoir of hepatitis C virus. It's just replicating in you, and it can do so for many, many, many years and cause liver cancer. But if you take an antiviral drug for as little as 4 to 6 weeks, you can get rid of it entirely. But many people at risk for Hep C get it again because they engage in risky behavior like intravenous drug use. So we do need a vaccine, but we don't have one.
And we'd also like a vaccine because vaccines are usually the better solution.
Yeah.
I mean, just in terms of expense and accessibility, you take it once and you're done.
Yeah, and these individuals who keep getting infected, they would get a vaccine and they could do all the drugs they want. They're not
Well, I mean, it's also just better to have prevention
Yes.
instead of totally relying on control, because then you can stop having some of the damage or prevent the damage instead of having to just sort of fix it or live with whatever damage happened during your infection.
Yes.
Unfortunately, we don't know a lot about antibodies and prevention of disease, which really surprised me because, as you will see in this paper, there's a lot of fundamental stuff that's discovered that I would say should have been known already. But it's difficult to grow the virus, and it had been for many years. But this is an enveloped virus. It is plus-stranded RNA with a membrane, and in the membrane are glycoproteins called E1 and E2, and these allow the virus particle to get into the cell. The E2 protein binds the receptor, and the E1 is involved in stabilizing E2 so that it's able to interact actually with multiple molecules on the surface of the cell in order for the virus particle to get in. But the real issue with hepatitis C virus is, like many RNA viruses, the genome mutates at each replication cycle. The RNA polymerase is highly error prone and there's no error correction. And so individual strains of Hep C can vary by as much as 35% of their nucleotide sequence.
I think even for a virus, that's a lot.
It is a lot. The genomes are classified into 8 genotypes, and within each genotype, there are subtypes. And when you do that, things are out of control.
I feel like I'm really glad that the people in the HCV field discovered all of this before they heard the word variant.
Yes.
Yes, right. Yeah, they don't talk about variants. They talk about genotypes and subtypes. So these subtypes have between 20 and 25% nucleotide sequence difference. The most common genotypes are 1 and 3. 1 is prevalent in Europe, America, and Far Eastern countries, and 3 is prevalent in South Asian countries. And the genotype, of course, have different E1 and E2 glycoproteins, and this is a challenge for vaccine design, right? You're probably not going to have a vaccine that's specific for one place, but rather one vaccine that will get everything. And so that's not very easy to do.
Right. And, I don't know enough about the structure of all of these proteins, but you really have to think, are there conserved epitopes with this broad of genetic change, or are those epitopes just not going to be able to be shared?
Yeah, and you may have to generate a vaccine that has multiple epitopes in it in order to cover all of these. But before you can do any of that, you have to know what the correlates of immunity are, because you need to know what you need to generate antibodies against.
That's right. Now, this variation also impacts the antiviral efficacy, because some of the genotypes are not very well inhibited by the drugs. So genotype 3 in particular is particularly recalcitrant to antiviral therapy, and so is subtype 3b. And a lot of this variation happens to be focused on the E1 and E2 genes, which is unfortunate, right? Because that's what you're going to target with antibodies, at least. And so when we talk about antibodies binding the glycoproteins, of course, there are the epitopes where there's short amino acid sequences where the antibodies bind and then may or may not block infection, depending. But there are also glycosylation sites in the glycoprotein, amino acids to which are attached sugars, glycoproteins. And the common one is linkage of a sugar to asparagine, which is called N-linked glycosylation. And the E1 and E2 proteins of the virus have lots of N-linked glycosylation sites. And these can impact antibody binding as well, right? As you may guess, if there's a big bulky sugar there, the antibody can't bind.
Yeah, they absolutely can. And I like how you described it as a big bulky sugar, because I think I didn't originally recognize how big and bulky these carbohydrates could be in terms of occluding the epitope. In other pathogens, sometimes this is referred to as the glycan shield, but it really can be a big barrier to antibodies detecting the epitope on the protein.
Right.
It
And of
also
course,
can be really easy to change without changing the protein.
right. So these amino acids can change. The asparagine can change. You can eliminate a glycosylation site. You can introduce one by a single amino acid change. Now, the other thing they look at in this paper, which they're going to look at which amino acids are important for antibody binding and neutralization, but they're also going to look at host genes that may be involved in antibody responses. And it's already known that interferon lambda 4 gene is important for interferon-based responses against hepatitis C virus, and also the major histocompatibility region can also determine whether you clear infection or not.
And there's been previous work showing that this interferon lambda 4 gene variations in that are associated with different responses to hepatitis C. So some people have a variant that they clear the virus pretty easily, and others have a variation of that. Sorry, didn't mean variant. A variation of that that or a haplotype that is associated with worse outcomes.
So what they've done here, they have samples from 60 people who were part of a clinical trial, and they have serum from these individuals, and they look at the ability of the antibodies in this serum to bind E1 and E2 envelope glycoproteins first by ELISA. Okay, and so they can say how much binding activity is present in each one. And they are using pseudotypes. They're not using authentic hepatitis C virus. They just have the glycoproteins produced in a retroviral backbone. And then they can also use the same system to look at neutralizing activities of these antibodies. And they do it for each of the 60 samples. So they have individual sera from each of these 60 people.
And the reason you do this with a pseudotype virus is because hep C is a pain to work with, right?
Yeah, it is a pain to work with. Even though you can work with it, it is a pain.
Go back and listen to the Charlie Rice episode.
Yes, indeed.
I was going to say.
And so, not every antibody that binds will neutralize, as we will see, but they have a number here that represents the fraction of that. And also they can get the sequence of the hep C genome that infects each of these people. I mean, these people were involved in a hep C related clinical trial. And so you can get, they got the genome sequence of 57 of these individuals. And so from that, they can tell that 54 of them had been infected with genotype 3A, and 2 were infected with 3B and 1 with 2B. So they decided to focus on all the people infected with 3A, just to minimize the other, because there's going to be other variations going on there as well. And then they can also look at the interferon lambda 4 haplotype of the host and also the HLA alleles and compare that to what's happening with antibody responses, right? And so they see a correlation between binding and neutralization, which is good because to neutralize, you should bind. So that's a good correlation to see. One thing that's interesting is this is a largely statistically driven paper. They do experiments, but they interpret them with statistics. So they're always talking about association, which is interesting. And what is interesting, they looked at a variety of non-genetic factors that might be associated with antibody responses, right? So they're looking at sex, cirrhosis status. So if you're infected for a while, you could develop cirrhosis of the liver. If you've had interferon treatment, how old you are, and how much virus you have. And the largest impact on antibody binding, which they've just measured, right? We talked about it, is your sex. Males had lower binding levels than females. That's very interesting. Just now on TWiV, Nels and I live streamed. We talked about a science paper where they're looking at longevity differences between males and females of many, many different species. Because as you know, we think that in general, females of the species live longer. But it's not true. Depends what species you looked at. In fact, birds, it's the opposite. The men live longer. So that's a really interesting story. It has nothing to do with this. But here, the sex has the largest impact. Males have lower binding. So in humans, females live 5 to 10 years longer than males. And so maybe it has to do with their antibody responses.
Yeah, so
among other factors.
During
Among other
2020,
factors,
we had Sabra Klein on TWiV,
Right.
who does a lot of really great work on sex differences in immune responses. And if you look at her work and a lot of the other work that people have done, in general, you tend to see slightly reduced adaptive immune responses in males in humans, and a bit more in females. Some of that is probably due to gene dose, and some of it is due to actual effects of the different hormones on immune cells themselves. But that sort of overall pattern of males having a somewhat weaker immune response is sort of generalizable through many systems.
It's
Couldn't
interesting.
Sorry.
Go ahead. It's okay.
Some people refer to testosterone as immunosuppressive or immunomodulatory. Is that what you're referring to, Brianne?
I would say immunomodulatory. So both testosterone and estrogen can strongly impact all sorts of immune cells. And so looking at sort of the data from the Klein lab and also on immune, we did a super cool paper, if you guys are into this, from Peter Broden, who looked at immune responses in trans men after receiving hormone and how the hormone changed their immune responses. And so I think that based on that paper, for example, I would say immunomodulatory.
Okay.
But there are a couple different things going on.
In some island populations of humans, there's no difference. It's really interesting. And then, as I said, in birds, it's reversed, but not all birds. In mammals, you can find mammals where it's reversed. It's males live longer than, and there's lots of factors involved. One of them is interesting. If you have a non-monogamous species where the male has to find a mate every season, they put a lot of resources into that, right? Growing all kinds of fancy feathers and doing dances, and that makes them live shorter. It's really interesting. But lemurs are, what are they? Non-monogamous. They're not. So they pick a different mate every season. But male and female lemurs live the same age. So there's no one answer. Okay, so polygamy can have a couple of different arrangements in the animal kingdom. It can be the males try to mate in the same season with as many females as possible, or it can just be that they don't keep the same mate from season to season. Yeah, right. But the point is, if you have to search out a new mate,
it may
a cost,
right? That may affect it.
But and also the amount of care that a female has to give to offspring can also impact longevity. So anyway, It's a very interesting paper. But
I just looked at the naked mole rat, which is number 1 in longevity, says that it's, from my quick Google search, monogamous, but they have a social hierarchy with a queen.
Yeah. So they do. So I actually spent a bunch of time this morning reading about naked mole rats, weirdly enough. And the reason is that there is a paper that came out in Science linking naked mole rat longevity.
Yes, with CGAS.
To CGAS, which is a protein I think about in my lab. So there's a paper that just came out. I was talking with a collaborator about it this morning, and we were sharing facts about naked mole rats.
They have a lot of interesting biology going on.
Yes.
Yeah.
Anyway, so males had lower antibody levels. And also cirrhosis and previous interferon played a role. Patients without cirrhosis or with previous interferon had lower antibody binding responses. Why that is, we don't know. It's just an observation.
Well, you can imagine if you had previous interferon treatment, that probably immunosuppressed you a little.
Yeah, what about without cirrhosis? It must have lower antibody levels.
Maybe
as well,
you have less inflammation. Or maybe
Yeah.
if you have some cirrhosis, you've got extra inflammation, and so you're making some more antibodies.
Okay, so those are non-genetic factors. And what about genetics? So then the interferon lambda 4 haplotypes, they looked at again. So these have been as Alan said, they've been previously associated with spontaneous clearance of hepatitis C virus and response to interferon therapies, which used to be used before the direct acting antivirals. So they looked at the association between 3 interferon lambda 4 haplotypes. Interferon lambda 4 null, interferon lambda 4 S70, and interferon lambda 4 P70. And they looked for correlations with the antibody response. And those patients that were interferon lambda 4 P70 had lower antibody binding than patients with interferon lambda 4 null. But the other two were similar. So again, there's an effect of these. It's a correlation, right? You'd need to do experiments of some sort to figure out causation, but it's a correlation. They wanted to look at the association between changes in E1 and E2 and the antibody response, right? And they again, it's an association because we're not doing experiments that would show it yet, but they try and rule out sex, cirrhosis status, interferon treatment, viral titer, which are all these non-genetic factors, to try and minimize confounding effects. And what they find is one change in E2 at amino acid 653, which was associated with antibody binding. So when you change that, you can impact antibody binding.
Yeah, and this is not to say that these experiments with correlations are not important, because they don't give you that sort of mechanism that you get after the fact. You need to know what the correlation is in order to have a hypothesis to test for your mechanism. So it's important that you know that the non-genetic factors aren't driving all of this before you do your big mechanism study and find out, it was actually all cirrhosis.
Yeah, so you would take this change and you could introduce it into a glycoprotein and see the effect.
Exactly.
Right. So that's just binding. That's just antibody binding, which is not. It's a good because antibody binding alone can have significant antiviral properties. You don't have to neutralize infection in, say, a plaque reduction assay. However, they did look at neutralization, and they found 2 sites, 501 and 533 in E2, which associate with neutralization sensitivity.
But their false discovery rate was 20%, which means that like 1 in 5 times it's going to be wrong.
Yeah.
False.
Right,
so
right. That's why you'd have to do the introduction study, right?
Yeah, definitely.
Yeah, so these are the sites that popped out on the top of the statistical graph. It doesn't mean that this is a lock.
Mm-hmm.
So 501 and 533 also associated with, right, so neutralization sensitivity, whereas a change to, whereas asparagine at 501 is associated with higher levels of neutralization compared to other residues. And I'm wondering if that has to do with glycosylation, right? Because it's an asparagine where you would be attaching a sugar. And also at 533, if you had a glutamic acid there, you got higher neutralization sensitivity compared to other residues. Again, 533 is not a glycosylation site, so I don't know how that would be working. Then they looked at the association between the changes in the glycosylation sites in E1 and E2 and antibody responses. Again, whether changes in these sites which lead to which sugars are attached correlate with antibody responses. And again, at 20% threshold, motifs at 3 glycosylation sites were associated with binding and or neutralization. And so NIT is one. It's site 476 in E2 was associated with higher levels of binding and neutralization compared to non-NIT motifs. That gives you, this is giving you a flavor for what they're doing here. How about the viral diversity within a patient, right? So if you do next generation sequencing of virus in a patient, you can get a sense of the population diversity. And so they use that diversity as a variable to test for associations between antibody responses. And in fact, an increase in patient in a particular part of the E1 and E2 genes was associated with greater binding.
Would it be fair to call those quasi-species?
I think so, yeah. I
Okay.
think that would be fine, yeah. Then they look to see whether these areas of the glycoproteins they've identified are structurally accessible and immunologically relevant, right? So they have they can map it onto the surface of the polypeptides. The structures have been solved of these, so you can ask: are these residues on the surface, or are they somewhere else? And so 653, which was associated with controlling antibody binding, it's on a highly conserved surface of E2. And then you have glycosylation sites. These are these have a lot of sequence variability to them. They're not so much conserved, but again, on the surface of the protein. And so these changes in the glycoproteins that they've identified, they want to know what if they do anything structurally. So they turn to alpha-fold 3, and they put these changes into the glycoprotein and have it alpha-fold 3 solve the structure, right? And of course, with the reservations associated with that kind of prediction, the changes don't make major conformational changes in the protein. So in other words, they're probably having local effects on antibody binding, not altering the structure of the protein grossly. They say the changes in antibody binding are likely driven by epitope-specific surface alterations, or glycan structure.
Which makes sense because the virus needs this protein in a particular configuration in order to get into cells.
Right. And they find that 3 of the 4 sites that they identified on the glycoproteins E2 are associated with neutralization are within or near binding sites of well-characterized neutralizing monoclonal antibodies. So these have immunological relevance most likely, and you probably should think about that when you're designing a vaccine, right? And they point out here again that these non-neutralizing antibodies that they find, they have other ways of being antiviral, which I pointed out, but I want to point out again. So that's the story here. We have an analysis of sites from patients that can impact and change and impact antibody binding. And if you want to make a vaccine, you need to consider that in the vaccine design.
Well, and everything that they did was more assuming linear epitopes, kind of what Alan was getting at. If they're conformational epitopes, then maybe that's being lost in this analysis, like to have more information about what's actually in contact, or which epitopes are actually closer than we think, or important for immunogenicity, etc.
I also should point out that these patients were chronically infected, right? So they say that this is really the first analysis of epitope changes in a chronically infected patient, not just in an acutely affected patient.
Right. And there may be something interesting to that in terms of are you making some kind of unique immune response
Right.
that allows that infection to persist as a chronic infection, and you didn't make some kind of response that would have cleared the acute infection.
So, Brianne, how would interferon lambda 4 polymorphisms affect antibody responses?
Well, I could imagine it in a couple ways. One of them is that the interferon lambda might change viral load levels.
Mhm.
And so the change in viral load could change the antibody level.
Right.
Alternatively, the interferon lambda might influence actual B cell biology,
Right.
and might change maybe class switching of the B cells or amounts of antibodies being made or some aspect of the B cell activation.
They also talk about ER stress, impairment of MHC presentation, right?
Yep.
Which has been observed with these interferon lambda 4 polymorphisms. So that could influence antibody response as well.
Absolutely.
Right? Yeah. Right. So they conclude by saying vaccines must address naturally occurring polymorphisms to enhance cross-reactivity against diverse variants and elicit robust humoral immune responses. And so the variants popped up in the last paragraph.
Yeah.
Actually, probably before that. Okay.
So, of course, I'm sure absolutely no one is surprised
No.
that I have a question of, well, what about T cells?
Yeah.
Yes.
Yes.
What kind of T cell responses are happening, and how would a vaccine that might influence T cell responses be useful here?
Well, also vaccine strategies, should they be or will they be more accounting for host factors? Like if somebody like screening for interferon mutations, etc., or even like a tailored vaccine, I guess, depending on people that are demographics that have higher incidence of having these mutations, should they be screened for these things? I don't know how feasible it is, obviously. Or even having, I guess, what we talked about at the beginning, multivalent vaccines, but that would be within subtypes, like within the, I guess subtype is the right word. Is that what we used? Like within the 3A.
Yeah.
I don't want to say variants, but like variants of the subtype, then you'd have to have a multivalent, which then I guess it gets a little bit crazy. That would be a lot of different vaccines, but perhaps.
Yeah, the problem is that if you do that, then, so the idea that one genotype or subtype is prevalent in one place is not absolute, right? There's always a little leakage.
Mm-hmm.
And then as soon as you have a vaccine against the one, then the other one is going to predominate because it's there because of a founder effect, right?
Yeah.
So if they say here, let's see what they say here in the beginning, we will. So they say one is highly prevalent in Europe, America, and the Far East. It's probably because that's the first one that went there.
Yeah.
Mm-hmm.
Diversified. But, if you put in 3, it would probably take off as well. It's just a matter of the founder. So I feel that if you gave Europe, America, and Far Eastern countries genotype 1 vaccine, then that's asking for genotype 3 to move in.
Yeah.
Right.
It just makes it a fitness advantage for genotype 3.
So I would like to make one that covers everything at once. Right.
Yeah.
Well, then you would need something that's like a functionally constrained site on HCV globally, if possible, and then something that the virus needs, right? That it needs to, like something it can't give up. You know what I mean? Like something
Right,
like functionally necessary for the virus. I just don't know what that would be for HCV or if that's even been identified. Yeah. Not like how it could mutate, but something that it needs no matter what.
And that's why T cell epitopes that are internal to the virus structure might be useful.
And they're likely to undergo less variation, right?
Yeah. But you could, depending on what we subsequently learn about the degree of variability in responses and in antigens here, it might be possible to narrow down the number of possible epitopes and say, okay, we don't have 1 target, we've got 4, but you could make a 4 antigen vaccine that would potentially do that. It's just a little more complicated.
Pamela Bjorkman, right? She's making these ferritin nanoparticles with many different coronavirus spikes on them. Maybe you could do something like that with E1, E2 from different genotypes and subtypes and cover everything. Right. All right. So our next paper is about a vaccine that exists. Nature Medicine, varicella-zoster virus reactivation and the risk of dementia.
I thought this
And,
paper was really cool.
Yeah,
is
I thought
cool.
this was really interesting.
Yeah, I had seen this paper before and was excited when I saw it and was excited to reread it and was talking to some colleagues who are now all trying to look for their vaccination records.
Right, so first author is Vitaly Polisky, and senior author is Patrick Schwab, and the group is from multiple campuses of GlaxoSmithKline, GSK, also Stanford University, the Chan Zuckerberg Biohub in San Francisco, and University of Oxford.
So this is about varicella-zoster virus, a herpes virus, so an envelope double-stranded DNA virus, which before vaccination you would typically first acquire in childhood, and you get chickenpox, which I got as a kid because there was no vaccine, and I
did too.
remember it.
I
Oh
did
my
too.
gosh.
I did too.
I remember it quite a bit.
It was big, big lesions, man. Some of them
Mhm.
really hurt. And then, you recover, hopefully, if you have a good immune system. And then when you're 50 and up, that virus has been with you your whole life, residing in some ganglia.
Because it's a herpes virus, it never goes away.
It never goes away. And it reactivates and causes zoster or shingles. And so now we have a chickenpox vaccine, and we have a zoster vaccine. And so there have been suggestions that reactivation of varicella-zoster virus, which is this process I just described, is somehow involved in dementia. And we don't really understand what reactivates varicella-zoster that much. Like herpes simplex, we understand the reactivation triggers for sure, like stress and UV light and so forth. But for VZV, we're less sure. But there's some indication that reactivation correlates with dementia. So what is dementia? A decline in cognitive function, mostly as you age.
Isn't
right?
that also just aging in general?
Well,
I'm joking.
right. So yeah,
To a certain
there's,
extent.
There's
To
a
certain
clinical
extent-
boundary for it when you get to the point of
I mean, even myself, I'm only 36, and I feel like certain cognitive decline at times.
No,
where
no,
before
no, you don't
I
have
would
any-
remember.
No,
no.
You don't have any cognitive decline.
No.
No way.
But you know what I mean with age. Like when somebody that's 40 compared to somebody that's 80, there is a certain level of like cognitive.
Hey, I remember all my
velocity,
viruses. It's a
speed,
matter of what
that's
you
true.
use. It's just that you don't use stuff all the time and you forget
is
it.
true.
I think it's a combination of what you use and also how much you have remembered in your life. Maybe you just run out of space.
But of course,
Right.
as you age, there's no doubt there's an increase in decline in cognitive function. Yes, for sure. And they say here the number of people living with dementia is expected to reach 153 million in 2050.
Yes. So I have a question. So I know that there are two vaccines that one can talk about here. The
Yes.
herpes zoster vaccine,
Right.
which is Zostavax from Merck, and that is alive attenuated. And then the recombinant, which
Right.
is
Shingrix.
Shingrix from
Shingrix.
GSK.
Right.
So are they giving both for, is one chickenpox?
So
and
far.
One,
So far, the Zostavax is for chickenpox, and Zostavax used to be, so they're trialing Shingrix for chickenpox now because it's so good at preventing shingles.
right. I mean, it makes so much more sense to do
And it's
Shingrix.
also it's a subunit vaccine, whereas Zostavax is attenuated, right? And we don't,
right. Well,
probably a certain number of them get latent vaccine virus.
that was exactly what I was thinking. Is to me,
Yeah.
it makes I feel like at some point, some of those people who get Zostavax are still going to have susceptibility to shingles. But if we could switch everybody over to Shingrix, then we wouldn't.
Yeah, I
be
think that's the goal,
inducing
isn't
any
it?
latent infections.
Do you mean
I think-
you're referring to children, Brienne?
Yeah, yeah.
Yeah.
The subunit? Okay.
Yeah, I think that's kind of the general plan in public health circles, and I'm sure it's the desire of multiple authors on this paper who are from GSK, the manufacturer of Shingrix.
As a person who just thinks about immune systems, that would be my goal too.
Yeah.
Okay,
Yeah,
see,
I got
before
Shingrix,
it was like
by the way.
Before, I remember everybody just wanted to get chicken pox at least once so that you wouldn't get it when you were older. But now it's like, no, you don't even need to get the virus necessarily. It would be ideal that you didn't. I remember, because I'm one of 7 kids, my brother got chicken pox and my mom literally said, go rub up against everyone so we all get it at the same time.
Yep.
And I remember doing that.
I very much remember it because I got it the year before kindergarten. And all of the other kids also wanted to have it before they went to kindergarten. So they wouldn't have to miss kindergarten. And so I remember feeling very poorly and having to have a tremendous number of friends over. And I did not want to see my friends. I wanted to, lay around and be sick. And that my friends just kept having to come over.
Yeah. So
I got it when my older sister got it.
Yeah. You just.
So I got shingles in episode 5 of TWiV.
You had shingles, like actually?
Yeah, I got it. So I had chickenpox as a kid. Then on episode 5, which was October 2008, I got, because it was Saul Silverstein was my guest, and we were talking about my shingles at the time. So now I have a timestamp for your shingles
your outbreak
episode.
Did you subsequently get vaccinated?
What? I got vaccinated. I wasn't 50 yet, I think. Wait.
Right, okay.
Let's see, 2008, how old was I? 53 it's too hard. I can't do the math.
It's cognitive decline.
Well, I never was very good at math, but I might have been, let's see.
So you should be 50. So you were born in 53, then you should be 55.
You were 55 in 08.
So you would have
I guess I hadn't
been around there.
I had not gotten the shingles vaccine. I should have.
Well, I think,
but then
hasn't the shingles vaccine age recommendation been coming down?
I think it
was
came
it
down.
50?
It's 50.
Was it 50 then?
I don't know. It's a good question. It may not have been. But that back then it would have been Zostavax. It wouldn't have been Shingrix, right? Yeah. And then Shingrix. I only got my Shingrix a couple of years ago. So. But the shingles, you didn't get shingles, Alan, right?
No, I got Shingrix. I had heard from enough people who had shingles to know that I absolutely didn't want that.
It was moderately annoying.
Oh, okay. I've heard much worse stories of shingles.
Yeah, it can be
It can be really, really terrible.
Anyway, back to dementia. We don't know what causes it, but there have been implications of that neurotropic viruses might be involved, right? Because they're going in the brain and they can do things there. We have some evidence that they're involved in dementia and Alzheimer's disease, herpes simplex viruses, varicella-zoster viruses, which pretty much everybody is infected with. VZV can induce a kind of Alzheimer's-like pathology, right? The deposition of amyloid peptides in the brain. You can find other markers of Alzheimer's in people infected with these viruses, and cells in culture. You can do experiments that are consistent with that. And in fact, in animals, if you infect animals with herpes simplex 1, you get increased accumulation of amyloid and phosphorylated tau, which is also implicated in dementias. So there's quite a bit of evidence that these herpes viruses are involved with dementia. And
there are 2 prior TWiV episodes in which we delve into that.
That's right.
So TWiV 1207, taking a shot at dementia, and 1249, demented and crass. So we have talked about this before.
And so studies have been done showing that treatment with antivirals against herpes can reduce incidence of dementia. So, for example, anti-herpes virus treatments, also vaccinations against VZV are associated with a lower risk of dementia, but we don't know how. And of course, there are always confounding variables. One of the things I learned from this paper is that in an observational study such as this, you can never prove anything. Because you can never eliminate 100% confounding variables. You would have to do a blinded double trial, and you can't always do that in people.
That is correct. It's like you're going to the next section of my epidemiology class.
Well,
but they do a very good job of trying. They do.
They do a good job. This is probably the best that you can do. And also the Epstein-Barr MS study that we did is also another example, which they cite here. I mean, there's some ways you can get almost good enough. But as they say,
Yeah.
you can never prove it like you can in a placebo-controlled double-blind trial. And I want to emphasize that because the studies on autism are always an association. They are never a causation. So for RFK and Trump to say, Tylenol causes this is absolutely wrong. It's fundamentally wrong at the highest scientific level, okay? Because those are observational studies. But if you do them in siblings, you get rid of a lot of the confounding variables, right? So if you have a mother has given birth to kids, one she took Tylenol and the other she did not, then you can compare them and you get rid of a lot of the confounding variables.
But not all.
Not all, never all, right? There's always something that's going to fool you. And maybe that's the one lesson you learn as a scientist. Well, that's somebody's definition of science is doing things to avoid being fooled.
Yes.
Yeah.
Yeah. So in this paper, one of the things I really liked here, by the way, both papers today are open access, is in the introduction, they lay out their hypothesis in this clear, very stepwise way. They're, if VZV reactivation contributes to dementia, that entails 4 assumptions, and they lay out what the assumptions are. You would see this, you would see this, and then they introduce how they're going to approach it, which is this massive database of electronic health records from 100 million people. So that certainly helps in a correlative study.
Numbers are good.
Yeah, the 100 million is awesome, and Alan is right. This part in the introduction where they go through sort of, if our hypothesis is true, here are the things that would happen. Here are the assumptions. Is really quite spectacular, and I also thought that was one of my favorite parts to read and see how they laid that out.
So let's go through it. The first assumption: if you have more VZV reactivation, you get increased dementia risk. They look at these electronic health records. They look at people with a single episode of zoster versus multiple, right? And so you should see an increased dementia risk. Second, that a lower burden of reactivation gives you decreased dementia risk.
It's the inverse of the first assumption. Yep.
Yeah, they look at people who got a zoster vaccine. This one they're looking at both Zostavax or Shingrix. The third assumption is that the modulation of reactivation is proportional to dementia risk reduction. So there you compare people receiving different types and doses of zoster vaccines, like 1 dose of Zostavax, 2 doses or 1 or 2 doses of Shingrix. And the fourth assumption that the impact of reactivation is proportional to protection. And there you can look at the decline in protection of the vaccine against shingles and see if that correlates with a decline in protection against dementia.
And there's a fifth assumption I miscounted. Sorry. So, and that is that people who are inherently at higher risk of VZV reactivation would get more benefit from preventing VZV reactivation. In other words, more benefit from the vaccine.
So these are all independent ideas, assumptions, and as you'll see, they all fit the hypothesis, right? Whereas if one didn't, then you'd worry that there's some confounder that you're missing in there. And by the way, speaking of confounding variables, they've identified 400 electronic health record derived confounding variables, right? And try and eliminate those. We'll talk about
that. Must have been an interesting brainstorming session.
Yes, right. Well, you can look at the records.
You can look at the records. Yes.
What else is going on?
What else might?
So this is the Optum electronic health records database. 100 million individuals from 7,000 hospitals in the U.S. between 2007 and 2023. I'm just thinking, when you finally finish this study for all these years, oh my gosh. Some of these people, so their records of reactivation were kept, obviously, and they got in their vaccination records. And then they construct a set of 400 pre-exposure characteristics. This includes demographics, comorbidities, lifestyle factors like if you smoke, cardiovascular conditions, diabetes, healthcare utilization, and healthcare seeking behaviors. That's really important because people who seek healthcare are different from people who don't, right? And they can have an effect.
And you also get different diagnosis frequencies.
Yeah.
Yes.
So they use this to try and remove covariables, confounding variables from the data. So they're looking at herpes zoster only, right? So that's this rash, which is the only reactivation form that they looked at. There are other things that happen. And they also used as a control vaccine the pneumococcal vaccine, the 23-valent pneumococcal vaccine, PPSV23, which doesn't protect against zoster. But a lot of these people got it, right? Because as you get older, you get it.
That was a good control, too, because you want to know, is it just that they got some kind of a vaccine juicing up their immune system that may have had an effect on their dementia rate, or is it specific? And so they included that. And I think those are, what is the pneumococcal vaccine age? Is like 60-something or...
I think it's 60, but I'm not positive.
Yeah, I'm not due for it yet, but it's coming up soon.
PPSV23.
They're both not things I can get yet.
Yeah, exactly. Two of us have had these
65,
experiences.
65.
65. Oh, okay. I'm a ways off.
Yeah, I've gotten. Well, I was told I got my last one because the latest one is you only need one. Okay, so what do we find? First, recurrent zoster is associated with an increased risk of dementia compared to a single zoster episode. Having multiple episodes is associated with a 7 to 9% higher risk of dementia at 3 to 9 years after the index date. That is the second zoster episode. So
and
after
they
your
define a zoster episode as to having a clinical rash, as a having
That's
like an
right.
actual dermal rash, as opposed to they're not taking into account any sort of subclinical
That's right.
infection, which is important to remember.
Yes.
Yeah, because it could be that those impacted also, right? So the incidence may be higher, the association may be even higher.
Yeah, and it's there have been studies showing that these subclinical reactivations occur pretty widely from time to time.
Yeah, and that's actually, I mean, there are lots of things I like about this, but that's why it's good that they look at not just that clinical reactivation, but also things like receiving the vaccine, which may prevent some of those subclinical reactivations and might be more useful than using an antiviral because you can do it as a preventative and maybe prevent some of those subclinical reactivations.
Yeah.
You don't have to wait for one that you actually see.
Yeah.
All right, next, vaccination against zoster was associated with reductions in dementia risk of 33% for Zostavax and 27% for 2 doses of Shingrix at 3 years after the index date compared to those exposed with the pneumococcal vaccine, right?
How often are you supposed to get Shingrix? Like post 50 years old? Is it every 10
It's
years?
2. It's 2
Oh,
shots.
every 2.
shots.
Okay.
2
shots,
and
but-
done.
And then that's it.
Yeah.
Shingrix.
Although, yes, that's it. Yeah.
And the second one kicked my butt. Not as badly as the COVID vaccine, but it was a little rough. But totally
So
better than shingles.
And this lower risk persists until 5 years.
And so maybe, as Angela is sort of, I think, alluding to, maybe at some point we might say, maybe you should get it more often
Maybe
Exactly.
get a booster.
reduce your dementia risk, even
I
if
was like,
it
wait,
may not
should-
reduce your...
Can
Because
we
one
just
of the
get it
things they're
again?
about to look at is the decline in antibodies with
Exactly.
one of these vaccines.
Exactly.
I'm spoiler alert, but I'll come back. I'll report back.
Now, two dose regimen of Shingrix has been studied, and 2 doses are more effective against shingles than 1 dose, right? We know that there are clinical trials that have done that, and so they compared 2 doses of recombinant with Zostavax, and that reduced dementia 18%, 18% lower for Shingrix at 5 years after the index date. And at 5 years, also, the risk of dementia was 24% and 19% lower in those who received multiple compared to 1 dose of Shingrix. So you should get 2 doses of Shingrix.
Yeah.
Okay, waning of protection. We know previously that protection against shingles goes down over time. And this is Zostavax, post-Zostavax. Shingrix hasn't been around that long, I think.
Yeah,
So-
Shingrix is newer on the market, so they don't have
know
a
that.
long enough time period to see whether there's a decline.
So in
But I
10
would
years,
assume so.
for 10 years, you can see a decline in 10 years for Zostavax. Prevention against shingles, right? So then they looked at the risk of dementia over a 15-year period after Zostavax relative to the pneumococcal vaccine control. The relative risk of zoster is stable, while the relative risk of dementia regresses towards the value of 1.
So I would say when I'm over 50, I'll probably get a Shingrix vaccine every about, 5 years? It looks like after 5 years, it can get a little bit hairy,
Little dicey,
and then
yeah.
suddenly a little dicey, and your dementia risk goes up. Also, because talking about subclinical infection, I don't want to say infection, subclinical inflammation or reactivation. Well, if it's these amyloid beta plaques, that is also that people have shown that VZV can contribute to. That's not something that's going to happen in like 2 weeks that you have a flare up where you see some sort of rash. That's going to be something that's over a long period of time. So it kind of leads to think that it is very frequent, maybe like these inflammatory, like controlled areas in a ganglia, that there's like a little bit of inflammation, and like tau, phosphorylated tau is accumulating slowly at like a very, very low level as opposed to these huge flare-ups. So maybe we should get Shingrix more often for
And maybe
dementia.
also earlier. Like maybe they should let 45-year-olds have it.
Yeah.
Or like 35-year-olds.
Now everybody's lining up for Shingrix.
But why not? Because I had chickenpox when I was, 12, 10.
Right.
And is it slowly doing that already in my brain, and the cumulative effects are only post 50 years old because it's so much time has passed and my immune system is maybe decaying slightly? I don't know.
Well, no,
And by the time you get Shingrix, maybe you're going to be as out of it as Vincent or me.
Imagine. Not joking.
well, no, but I think that the thing that I think is actually important about that is that you could imagine in an older cohort, they probably had more chickenpox exposures, more VZV exposures throughout their life because they were around other children who were getting VZV. And so each of those exposures boosted their immune response.
Right.
While someone who is in a younger age group got chickenpox and then hasn't had those sort of community boosts by being around other people with chickenpox, and so their immune responses may actually be decaying faster.
Yeah.
Good point.
Interesting to think of. Elementary school teachers are more protected, I guess.
Yeah, elementary school teachers, older siblings,
Older siblings, yeah.
things like that.
But chickenpox has also become rare because of the vaccine.
It's
Right,
true.
exactly. So I can't think of a time when I've been around someone with chickenpox.
Right.
But if I think about like my mom, who is an elementary school teacher who also had kids who had chickenpox, she was exposed many times.
Yeah.
I guess like in a nursing home, a lot of elderly people will have shingles. So if you're working in a facility where there's a lot of elderly people that could potentially have shingles outbreaks.
I'm just trying to rationalize why I should be able to get
Why
this
you should
vaccine.
be able to get this vaccine.
Can you
yes.
just pay for it and get it?
Maybe?
Is that a thing?
Well, when I got it, I scheduled it at a CVS, and they ask you how old you are.
Yeah.
Right?
You
Yeah,
could just say,
You'd probably...
nobody would believe Brienne.
I'm here for my Shingrix. Yeah. Put on a white wig or something. Yeah.
Well, I don't know if they check it. That's a question.
Yeah. I don't recall getting carded. But
No, I mean I just
I did
checked
sign
in.
up under my real name. Yeah.
Yeah, so I don't know.
Anyway.
Well, anyway, this effect, the long-term effect, they think it's because, the vaccine prevents zoster, but you're delaying dementia. So basically, when the vaccine effect wears off versus zoster, you have more reactivation. Therefore, the dementia is going up again. And they didn't do this for Shingrix because it didn't have market
They didn't
entry
have 10 years.
2017. Yeah.
No data.
No data.
Not
Okay.
enough data.
What about people who are at higher risk for herpes zoster? So older age and female sex are known to be associated with a higher risk of zoster. So if reducing reactivation of VZV is the mechanism for dementia, then you would see a greater impact of vaccination on progression to people with a higher risk versus a lower risk, right? So they looked at that and they found greater relative reductions in the risk of dementia for older adults or female individuals than the overall cohort exposed to vaccines compared to the pneumococcal vaccine at 3 and 5 years after the index stage. So the females over 50 with Zostavax had 35% and 31% lower risk of being diagnosed at 3 and 5 years compared to 33 and 27% for all individuals. And females 80 to 89 years old had 39% and 27% lower risk of dementia compared to 27 and 17% for all individuals. So it's a substantial reduction in the risk. It's pretty cool.
Yeah.
Then they looked at these 400 covariates. Of course, they couldn't do it themselves. They needed to use machine learning to ask what's the risk. And so what they do, this is an interesting computer experiment, they synthetically shift exposure dates by 1 year before the actual exposures here. So basically it's just changing the time of exposure, and any other difference in the cohort is remaining the same, right? So doing that, they found little evidence that confounders would change their conclusions. They got the same conclusions.
Right. So basically, the two groups are the same if you change the exposure date so that the exposure date can't be the thing that was driving the difference. And so that tells you that the exposure was the thing driving the difference, because when you take it away, there's no more difference.
Right.
But they did find that there's a higher risk of confounding when comparing vaccine-exposed to non-exposed cohorts, right? Which they say highlights the importance of extensive confounder control, which they achieved by these 400 covariates. And the cohorts are big. So those are the data.
Yeah.
It's a very consistent link between VZV reactivation and dementia on multiple counts, as we have said, and multiple vaccine doses are better at decreasing dementia risk.
Yeah, I really like this part that Vincent mentioned at the end as well, because as it points out, there's a difference between the people who ask for a vaccine and those who don't, and that's going to confound this risk.
Yeah.
And so the fact that they've actually figured out, controlling for that as well is really nice. I kind of was wondering as we were talking about it, who the people were who got a Prevnar, the pneumococcal vaccine, but didn't get a Shingrix.
Didn't get the Shingrix.
I'm sure, like, who are you? What happened to you to make this happen?
Inconsistency. Some people are more sporadic about their health care. Others are more diligent, and you ended up getting one and not the other. Either that or because the pneumococcal vaccine is given at an older age. Maybe they started taking their health more seriously as they got older, but not seriously enough to get the Shingrix.
Or they forgot Shingrix existed. Somebody suggested the pneumococcal, and they were like,
because the dementia was already setting in. Yeah.
That's true. 65.
Yeah.
No, I'm joking. I'm joking, Tom and listeners.
So they say we don't know the mechanism of reactivation effect on dementia, but we've already talked about how it could induce amyloid deposition. But it also turns out that when you have a VZV reactivation, that can reactivate herpes simplex virus as well, which is also a risk factor for dementia. And there are other things that VZV does besides shingles. It causes vasculopathy, which has been linked to cerebral events like ischemia and infarction and hemorrhage, right? That could give rise as well. And as Angela mentioned earlier, there's subclinical VZV, which could also impact dementia progression as well. And these were apparently reported by astronauts who go up into flight. They get subclinical VZV reactivation, so they shed in the absence of, of symptoms. So that would be an interesting study to do.
How does the astronaut risk of dementia? You're not going to get 100 million people on that one, though.
No.
So this is very
not
interesting
anytime
now.
soon.
The primary limitation of this study is that causality cannot be established without a prospective randomized controlled design. Okay? Confounding is a major concern with any retrospective analysis, and we cannot exclude the possibility of residual confounding in our study. I have to say that it's true for any study. And by the way, they point out the risks, confounding variables they didn't take into account are educational attainment, cognitive reserve, and physical activity. But they already knew that these are correlated with healthcare utilization and comorbidities, body mass, which were all controlled for. So they feel that that wasn't an issue.
Yeah, because those are things that would not be in the electronic health records.
That's right. Yeah, that's exactly right. So there you go. It looks really good, and I'm happy I got my 2 doses of Shingrix.
Likewise.
Sign me up.
But, it's not going to eliminate all dementia, right? No. There are other causes as well.
And it's not going to eliminate the normal cognitive decline that Angela was talking about, where all of us get a little bit worse with age.
Yes, but if you podcast on Microbe TV, you will delay the cognitive because you will stay sharp. Actually, some good ways to prevent cognitive decline include exercise and using your brain.
Yep.
Yes.
Mm-hmm. Don't watch TV all the time. So many people I hear are just watching, because now you could stream everything and watch a whole season in one night, right? No, don't do that. Read a science paper. Listen to an episode.
Walk.
Go
for a walk. Walk in nature.
Apparently walking in nature is good. Yeah.
Apparently, yes.
So there you
Imagine that. We lived in nature for thousands of years, and somehow now we think it's positive to walk in nature. This is the abnormal environment that we live in, not nature.
I don't know what is abnormal, Angela. There's just things, right? It's part of evolution.
New, and the environment we built for ourselves is normal to us because we built it. But still, we should get outside occasionally.
Anyway, this is the third in, as Alan mentioned, TWiV 1207, taking a shot at dementia, and TWiV 1249, demented and crass, the relationship of herpes viruses and dementia. Very interesting. I never thought this would be an issue. I never did. So there was a study at Columbia, which is done, where they took people with early onset Alzheimer's, determined by MRI, I think, or functional MRI, I don't remember. And they treated them with daily antiviral for a couple of years, and there was no delay in the progression, which they say probably means you have to do it earlier.
Yeah.
So once you have onset, it's too late.
Damage is done.
Yeah. Which is sort of why the vaccine is a nice idea, is you can get to that vaccine and actually get prevention before the damage is done.
Yeah.
Like in a 45-year-old.
Yes. Or 35-year-old.
Or 35.
And by the way, the cognitive decline thing reminded me some little bit of trivia I learned a few years ago. The mandatory retirement age for air traffic controllers is 56.
Wow.
Because of cognitive decline.
There you
go. Think it is normal cognitive decline?
It's normal. The data suggests that when you get up to your 60s, you can't maintain the level of alertness that is necessary to navigate thousands of people around in 3-dimensional space at 500 miles an hour.
But that's not contributing to the dementia, is it?
No. It's probably preventing the dementia, the fact that they're doing this.
Can't they get computers to do it for them?
Oh, no, God, I wouldn't want to be on a plane with computers doing the air traffic control. They do have a lot of computer systems, but not to do the actual, you want people watching that.
Well, you could use it as a help, as an aid.
Of course, yeah, and they do. And in fact, my mom's job for much of her career was she worked for a company that was overhauling the U.S. air traffic control system. It's the most complicated airspace in the world. And it was this massive thing, replacing old vacuum tube computers with modern systems.
Alan, these are federal employees, right?
They are, and it's a problem now with the shutdown. And yeah.
People think about this stuff.
The people doing this are not thinking about it
No.
until the consequences arrive.
I mean, there are many activities carried out by the federal government that really should not be shut down.
Yeah.
Correct.
Right?
Like the CDC, 600
Yes.
people being
that
fired
too.
overnight.
Yeah, that too.
Have you
things
seen
like
the
that.
messages on some of the websites? They
Yes.
switched the messages last week.
No, tell us.
So the messages used to say, and it depends on the website. They used to say something like, this website is not being updated
Mhm.
due to the government shutdown or whatever. And some of the websites, one of them is CDC because I was on it pulling something for one of my classes when I saw it and thought it was weird. And then I found out it was a news story that like this was happening more broadly. So they've been changed to say because of the Democrats shutting
Yes.
down the government. And so there actually is they're showing a video on that front in some airports.
Yes.
And on some of the websites, you actually have this like partisan message. It's so weird.
It's so ridiculous.
Yeah. In fact, many airports are refusing to show the video that's being shown at the security checkpoint about this because it's political.
Yeah, they're
Which
now
is,
suddenly
and it's
these
a really
super partisan things on these.
It's a
websites.
hilarious claim that the Democrats,
Yeah.
who control no branch of the government, are somehow responsible for this.
Yes.
But I understand what the argument is, but it just doesn't make sense on its face.
Well, they're depending on people not thinking
Not
about
understanding,
it and
yeah.
just,
Mhm.
oh yeah, it's their fault.
Right.
All right, we have a couple of emails here. Let's knock them off. Angela, can you take the first one?
Yes, Howard writes, I'm constantly informed and inspired by TWiV and other microbe TV efforts. Thank you. I'm reading Steven Pinker's excellent book, Enlightenment Now, as an antidote to the repetitive world is going down the tubes media reports. As a psycholinguist who wrote a book on past tense, he notes his favorite past tense sentence came out Wikipedia. Smallpox was an infectious disease caused by either two virus variants, variola major and variola minor.
That's a good one, right?
Was an infectious disease, yes.
Yeah. In reading this, it struck me that this landmark human achievement deserves a yearly formal day of recognition and celebration, and that the gang at Micro TV would be ideal to spearhead the campaign for this to happen. I see several benefits to this, among which would be reminding people of the benefits of vaccines, like the papers we explained today, reinforcing the need for supporting basic and developmental biological research, inspiring students to consider a career in biology and other sciences, recognizing the many thousands of heroes that made it possible to remove this scourge.
Scourge.
Scourge. Researchers, politicians, physicians, bureaucrats, nurses, community leaders, etc. Acknowledging that hundreds of millions of people have been spared a horrible death, providing a focus for concerted education and public awareness efforts on the value of investment in public health measures that yield invisible non-events that enrich and lengthen our lives. There are a bunch of days to choose from: the last death, the last case, the launch of the eradication program, the declaration of eradication. Howard, Toronto. P.S. It would be wonderful if you could get charitable status in Canada. You might want to poll your Canadian donors and followers to see if it would be worth the cost and effort. I suspect that it would be. I didn't understand the end. There are a bunch of days to choose from.
That you could
Yeah,
commemorate
the
as
last
for
okay.
celebrating.
Yes.
Like you could, yeah.
I would say because the only one I know off the top of my head is December 9th, 1979, which was like the day WHO actually eradicated it.
Yeah.
That's what I would pick.
I
am ready to celebrate December 9th.
Yeah.
Unless there's something else already celebrated. Let's look.
Or you could pick anniversary of Jenner's publication, or you
Oh
could
my
pick,
God.
Yeah.
December 9th is International Anti-Corruption Day.
It could also be International Anti-Smallpox Day.
Yes.
Good
Let's do
Lord.
it.
And we have so much corruption here in the
Yes.
U.S.
But we don't have smallpox.
Yet.
We don't.
Who
Yeah.
knows?
Okay. Yeah.
With how things are going.
Yeah.
Who knows?
I don't know. I don't know how you would make a day like this. I have no idea how you do it.
I think first you would make a flyer and post it on social media.
Yes.
I mean,
That's all you need to do nowadays.
That's all.
Who ends up declaring it? Is there a authority who declares?
Legislative bodies can declare something a day honoring something. So
Congress?
yeah. I mean,
But I think
once they
before
actually
that-
start doing their job again.
Yeah, I was going to say before that, I think just the internet.
Yeah, the internet's the place to start.
Okay. I think it's a great idea.
Yeah.
Just let it go viral.
Yes.
Literally.
You heard it here on TWiV.
Well-
And thank you, Howard. That's
December
a great
9th,
idea.
Anti-Smallpox
December 9th.
Day.
Alan, can you take the next
Smallpox
one?
Eradication Day.
Yes.
Gene writes, I came across this quote that fits the TWiV personalities. It was in the newsletter by Kay Jetalina, your local epidemiologist. She had a quote from Amit Kalantri. Systems fail when people with ability don't have authority, and people with authority don't have ability. Just guess who this refers to. You are our light in this darkness, Gene. Thank you, Gene. That's good.
Quote. I like this quote.
People with ability don't have authority, and people with authority don't have ability. That is a real problem in the world.
And Brianne, you're last.
Toby writes, Dear Twivers, my name is Toby and I'm an undergraduate biomedicine student at the University of Melbourne. I wanted to share with you a Reuters article describing the start of a Gavi-backed Salmonella typhi vaccination drive in Bangladesh, which aims to reach 50 million children between 9 months and 15 years of age in a 1-month timeframe. The vaccine is a typhoid conjugate vaccine, TCV, which is being provided free of charge with a particular focus on urban slums and rural regions, and which will be included in the Bangladeshi immunization schedule from November 13th. This vaccination drive comes in response to detection of multi-drug resistant strains and particular concerns around ceftriaxone resistance, given that ceftriaxone is one of few mainstay treatments remaining. This just goes to show that whilst waning vaccination rates in some parts of the world may reflect the spread of mis- or disinformation and people taking the benefits of herd immunity for granted, much of the world still has a large appetite for real science and the protection it provides. Thank you for all the work you do. From 10,500 miles away, your voices have a truly global reach. And Toby gives a link to the Reuters article.
I love it. I think it's great that they appreciate vaccination, right, and what it can do for public health, right?
Mm-hmm.
And in our corrupted society, it's just, well, you know what I'm saying.
Yep.
That's why you need Anti-Corruption Day.
Yes.
Yeah, you do. It doesn't. I've never heard of it before. It doesn't get much publicity.
Yes.
All right, let's do some.
It doesn't have the budget.
Doesn't have the budget. Yes. Let's do some picks of the week. Angela, what do you have for us?
So I have this Nature article by Lei Zhu called Am I Redundant? How AI Changed My Career in Bioinformatics. So I recently, I guess this is just like kind of a message to mostly graduate students, I would say. I mean, any scientist using now generative AI, be that Claude, ChatGPT, Grok, whatever you're using, that a lot of people think that, they're going to take over and it's going to take over our jobs in bioinformatics because these machine learning algorithms are going to be better than us. But one of the things that we have to remember, and this article states, is that it's true that you can do a lot of analyses a lot faster, some of them, but especially graduate students, this is like a warning, that if you don't actually understand how to do let's say sequencing, like if you have a bulk RNA sequencing data set, and if you don't understand like the basics of how to filter first and how to use certain thresholds and statistics and all of these things, the AI is only as good as you are. So if your prompts are terrible, your output is going to be pretty bad. And the thing is that a lot of people, like somebody that I know recently was analyzing their RNA-seq, and I said, you can use the help of, Claude. Personally, I prefer Claude to ChatGPT because it's like paid Claude is just a lot better. And it has better reasoning, especially with like scientific
It has better
ideas.
privacy controls too.
Privacy, exactly. A lot of it is just optimal. And ChatGPT, I just don't I don't use it anymore. Anyway, so this person realized that the code that was given to them was wrong and that they were misinterpreting their data and everything was inverted. And I looked at them and I was like, but you know, that's your fault. Like, I was like, that sucks. And it's been a month and a half and they've been building on these analyses and things like that. And I was like, yeah, but you need to check every single thing, any output that you receive, the code, you have to go line by line and make sure that it's correct. And I know this seems like common sense, but we instill too much trust into generative AI now. We think that it's perfect and that it knows more than us. No, even this morning, I was like, doing some exploratory analyses in my RNA-seq, and it was making all these like crazy claims about just things that it make absolutely no sense. And you call it out and you'll be like, actually, no, that actually biologically doesn't make sense because of X, Y, Z. And it's like, oh, you're right. So it's more like a warning to check our work, as everybody should already know, and to use it though, right? Because it can help do a lot of analyses a lot faster than you manually trying to obviously write the code manually. What I do is I have like in my R script, because I use R, I have like, let's say I want to do like differential gene expression analysis. I'll have it all written down. And then I'll say, okay, now I want to do this other comparison or this other thing. Look at this R script that I've already written, use all of the same thresholds, use all the same parameters, everything else, and now add this other thing. So I can do analyses really fast without having to write it all myself. And then if something seems interesting, then I'll go in and check line by line to make sure that it's all correct. So we have to learn how to use it better and to have better prompts, like what is it called?
Prompt engineering.
Prompt engineering, exactly, is very, very, very important. And not only that, the output, please, everybody, look at what it's giving it to you. We are still very, very valuable as scientists because we understand the biology. It has no idea what it's talking about. Like this morning, it was telling me that one of my findings was, it was amazing. It was going to be a Nobel Prize. And I was like, what are you talking about? That's an artifact of sequencing. Like it had no idea. It has no idea. So yes, we're still relevant.
I teach a first year writing class.
Gosh.
Yeah. So I have to teach I teach a first year writing class. And so I actually have to spend a fair amount of time talking about ChatGPT, and doing some things to make sure that my students just don't give me essays that ChatGPT wrote. So we have a lot of ChatGPT conversations. So there's some really great resources about all of this out there. But one of the policies that one of my colleagues came up with that I have absolutely stolen, with his permission, of course, is that he says that you should treat any output from generative AI like it's something your friend told you.
Yeah.
Mm-hmm.
So just like you wouldn't publish something your friend told you without checking into it more, you shouldn't go forward with something that generative AI told you without checking into it more. So you should think of it as, the rumor that your friend told you.
Like fact check, just fact check everyone.
Exactly.
Yeah, because it's the same thing in a paper. A paper is written and it has all the references, which we are literally fact checking throughout. So I don't know. I just think that people are getting kind of lazy where they just want like the output. Oh, this is fine. And then they'll just take it. But then somehow a month later when they realize it was wrong, they're angry at ChatGPT. And I'm like, no, you're the problem. It's increasing your productivity, but you at least need to do your homework and make sure that what it gave you is correct. Like that's the minimum.
For sure.
Anyway.
It'll give you an answer fast. It may not give you the right answer.
Exactly.
Right.
And then sometimes you go down a rabbit hole on something completely fake because you just didn't check, which waste of time.
Here's how I use AI. I make the thumbnails for the YouTube postings of all our podcasts, right? So last week we did an episode on, we had two papers on virus structures, right? An archaeal virus and a bacteriophage, beautiful tailed structures, right? But in the paper, and the title of the episode was Must See or Must Watch TVs, right? Tailed viruses. So I wanted to put both phages on a television screen. But they were all labeled with lines and letters and stuff, which would take me hours and hours to remove by hand. So I put them both into Google's AI and I said, get rid of all the lettering. And in 3 seconds, they were perfectly gone.
Yeah.
Perfect. I mean, you couldn't see any trace of where the black line had been erased over the capsid. And I said, okay, put them both on a television screen. Bloody perfect. A beautiful flat screen on a wall in a room with a nice potted plant over here and a remote control. Oh my gosh.
Perfect.
So
Good.
it was beautiful.
Good
right?
use.
So that's a good use, right? When you
Yes.
want to make an image. Or I'll say the one with the two chickens that were,
Yes.
I said, put them on the same gray background. It can do something
Yeah.
like that to fill the screen.
Mhm.
So you have to know what your use is, right? That's the thing.
Yeah.
I'm very happy that it just took all the lettering out perfectly. It was
I
so
love
cool.
AI transcriptions. Totally changed
Mhm.
my workflow.
Yeah.
But,
Yeah, this-
I'm going through the transcription to quote somebody, and I read the transcript, and I will pay attention to what they actually said as
Yeah.
well. And I have the recording, which I'll go back to and say, did they really say that? Oh, not quite.
Yeah, you have to check it.
Mhm.
Yeah.
Well, this program we're using now to record, right? It's AI driven. It's doing a transcription, right? And you can get the transcript of the whole
Yeah.
episode and who said what because we have our names put in. But
Mhm.
also, it will make 1 to 2 minute clips of what it thinks is interesting, right? It
Which is
says ready for
hilarious.
social media. And most of them are just useless,
Right.
and we make our own anyway. But it's interesting that they're trying to do that.
Yes.
Yeah.
Okay, Brianne, what do you have for us?
So I have mentioned previously going to some planetarium shows at American Museum of Natural History, which I've really, really enjoyed, where they have been doing some amazing visualization on the planetarium dome. And they use a software called OpenSpace, which has been designed by NASA and NSF, and is actually downloadable along with much of their data. So if you would like to scroll through the universe and actually explore data on the universe yourself, you can install this software and sort of make your own planetarium visualizations to your heart's content. If you go through the website, they have some videos and some visualizations already made. They've got recordings of some live streams of using this at planetarium shows. And they also have a link to their YouTube channel, which I've also linked, which is, again, some videos made using this software. And so every time I've gone to a show that has involved this software, I've been in total awe and loved it so much. And so I have started learning how to play around with the software myself and watching some of the videos they have put up. And so if anybody wants to see some of these really cool demonstrations of like everything in the universe, like you can fly through the whole universe, here is something that your tax dollars have actually supported to let you do that.
And it's as a result of the taxpayer support, it is open source. They give one of the other links they give is their GitHub page if you want to really, really dig into the software.
Yeah,
This is cool.
It's really cool. They talk about all the people who've, got PhDs and master's theses from it and things like that. And it just seems awesome. It's cool to play with if you kind of like space on the side like me, and if you are a data visualization kind of person, seeing how they've taken this to visualize the data is pretty cool too.
Neat.
I'm definitely downloading this. This looks so cool.
It's really cool.
It's done with federal money.
Yep.
Just saying.
I mean, I'm not American, so I'll just freeload, I guess.
Yeah.
No, that's not what I meant.
No, I know. I know. I'm just being lighthearted.
Just saying that this is
One of those
what's at-
many services that we should keep going.
Yeah, so should not be put in jeopardy because of bean counters wanting to redirect funds and destroy
It's not the
things.
bean counters in this case. It's the politicians.
Well, Russell Vogt is a bean counter in my
Okay.
view, right? All right, Alan, what do you have for us?
So this is a pick I've had sitting in my list for a while, but I go back to it occasionally. It is a live camera on YouTube that is underwater in a harbor along a busy shipping lane, which you wouldn't think would be all that interesting. If you did this in New York, it would definitely not be interesting. But this is in Miami, in Biscayne Bay, and it's sitting on a coral reef in one of the busiest ship channels in the country. And what's really cool is, despite the fact that this is an urban commercial harbor, there's this reef full of life. And so I'm linking to the camera itself, which you can tune into. And see, right now we've got a bunch of smaller fish swimming around here and just very, very active biologically. And there's also an NPR article that I'm linking to about the origins of this. A guy who's a young marine biologist who set it up because he was curious about what's going on with the reefs in such a busy place, and it caught on.
This is very cool. It's very soothing.
Yeah.
This is nice.
Obviously, there's a current here. They're all swimming into it.
Yep.
Mm-hmm.
Yeah, it's a tidal basin.
I have a feeling this camera is going to become something I see a lot of.
It's also got noise. If you turn the noise on,
Yes, you can get the water sound.
You can hear also is it a boat? I heard like
must
a
be some,
Yeah.
like
some
a motor, probably.
Yeah.
But this thing is sitting on the ocean floor,
It's
right?
sitting on the reef.
On the reef, yeah.
So it's underwater.
And it's broadcasting Wi-Fi, I guess.
I guess he's got either that or he's got a cable to the surface. Wi-Fi is not going to get through salt water, I don't think. But he
must have
cable to the
hardwired,
surface, and
right?
then-
Yeah. How cool.
So instead
Very
of some
cool.
aquarium channel, you can put that on. I haven't tried putting this on for the cats. That might be something.
They might really like it.
So
Yeah.
I guess at night you can't see anything, probably, right?
It probably gets kind of dark.
Except on a full moon, you might be able to see
Maybe.
things. Oh, that's very cool. That's
Very
just great.
cool.
We'll have to check.
Yes.
I wanted one really deep so I can. Oh, look at a grouper now just showed up. I want to see one where we can see giant squid and sperm whales.
Right.
That would be very deep. Well,
Those
sperm
don't
whales,
come
they
by
come to
very
the
often, though.
surface.
Yeah.
True.
Yeah, I know. That'd be too deep for a camera. It would probably crush it. Oh, this is neat. So in New York, it would just be black, Alan? Nothing there?
It would be, well, the visibility in New York Harbor is really crummy. All along the East
The
Coast.
pollution, yeah.
It's not just pollution. It's also it's the North Atlantic, which is a lot of algae, a lot of
Like silt.
lot of silt in it.
Mhm.
It's not as clear as tropical waters, just inherently.
This is
Mhm.
basically a 24-hour live stream.
Yeah,
And people are making comments here. It's so funny.
yeah.
Another jelly comes from the bottom left. Getting full narration. That's pretty cool. Nice. All right. So here's some self-help videos for you. So for a couple of months, my neck has been bothering me. And right now it's great because I'm doing these exercises from Dr. Adam Fields. Neck pain relief exercises. And he says, if this is helping you, please share it. So I'm sharing it. My social media person, Kim, recommended these months ago. And I don't know why my neck is stiff.
Maybe it's the
because-
magnesium.
I started taking magnesium lately, and it's gotten a little better, too.
Oh, okay.
Is that really what might be part of it?
No, I would just, you mentioned last week, I think you mentioned magnesium because you were thinking of taking it for sleep.
These are 10 minutes every day I do. I
Okay.
repeat them. It's seven days worth, and you repeat them. And it really helps a lot. So if you have neck problems, you should try these. Now, he's very corny. And in any other world, I would never listen to any of this, right? He says, motion is lotion. Movement is lubricant. Shit like that.
Stuff like that. Stuff like that. Yes.
We're stimulating production of brain neurotrophic derived growth factor. It's stimulating new connections. No, you're not. But, he says all this wrong stuff, but the exercises actually work. And, I feel better. I'm not cured, but it feels better. I can actually turn my head without pain right now. This is unusual.
Cool.
Did you change your pillow, Vincent?
Oh, God, I went through 4 pillows. It didn't help.
Oh.
I spent a lot of money on pillows. I have one now that's got all these grooves and stuff. And that's
Holds
the
your
worst
head.
time, actually, sleeping. In the morning when I get up, it's the worst. It hurts more than. And then as soon as I'm up and I'm moving around, it gets better. And then a warm shower and these neck exercises really help. But the best is distraction, like doing a podcast. I don't even think about it, and it's fine. But these are really good. And a lot of people have neck pain, apparently. So check out Dr. Adam Fields. He's a chiropractor on the northwest of the United States. I've linked to episode 1, but you can You can find all the other ones there. And we have a listener pick from Greg. Below are 2 suggested picks of the week, which are in response to the letter from Terry about AI read in episode 1261. Terry characterized AI as an elaborate autocorrect program, which I think may be accurate for the current publicly available AI models, but there is an enormous amount of money and brain power being invested to increase AI capability. Nobel laureate Professor Emeritus Geoffrey Hinton, a.k.a. Godfather of AI, recently gave an interesting and entertaining interview on the history and possible future of AI in conversation with Jon Stewart, formerly of The Daily Show fame. Professor Hinton described Jon Stewart the smart student in the front row who doesn't know anything but asks good questions. The conversation delves into the background of how human brains function, the development of search engines, and various steps along the way toward more advanced AI models. Going beyond autocorrect, Professor Hinton says that when the new AI models are being tested, they can pretend to be dumber than they are. 1 hour and 9 minutes into the video, which creates a problem for developing AI models that will honestly cooperate with humans. Isn't that great that they're lying to us?
That is not great.
I have zero expertise in AI, and so I cannot vouch for the accuracy of this conversation, but Professor Hinton seems to have highly appropriate credentials for this topic. Perhaps correspondents with some expertise in AI could critique this conversation or will suggest a better overview of AI that is more relevant to virology. While I was writing the above, the October 14th Harvard Medicine News arrived with an article about using the AI model Dr. Kabat for medical diagnoses, link below. This is much shorter and perhaps less entertaining than the 1 hour, 38-minute conversation between Jeff Hinton and Jon Stewart, and may be more appropriate for a clinical update episode with Dr. Griffin. Please share as you see fit. Thank you very much for your valuable science communication work. Greg is in Champaign, Illinois, and provides links to these videos and articles. I just remember the pick of Brienne's, which said that physicians who use AI lose their diagnostic capabilities.
Yes,
Remember that?
I have had that conversation and had my students read that same article.
It's good. I like that very much.
That same article, since I have looked at it recently with my students, has a link to another study from MIT where they actually looked at college students and did MRIs to look at changes in their brain and how they were learning with and without it.
Well, we do these podcasts without AI pretty much, except for the thumbnails now. Someone said, Vincent, it's a slippery slope. No. I'm not going to use it for anything else. That's all. Actually, we're all bots here.
Right. I was about to say maybe we're fake. It's getting pretty good.
Catch the wave.
I don't know, Angela. I think the plant behind you is fake, but it's real.
No,
apparently.
It's real. I have a dead leaf. I showed you the dead leaf.
Oh, that looks real.
I have to cut it off. It's real. It's real.
Well, that's episode 1263 of the AI-free TWiV. You can find the show notes at microbe.tv/TWiV. You can send us questions or comments or pics to twiv@microbe.tv. And if you enjoy these programs, we'd love to have your support to continue them. As you know, we don't do ads. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Angela Mingarelli is at McGill University Immune Vet on Blue Sky. That's IMMUNEVET. Thank you, Angela.
Thank you. I had a great time.
Brianne Barker is at Drew University BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.